Hello, everyone. Good afternoon. We're going to give it a few minutes and get started soon. All right. Hello, everyone. Thank you so much for joining us. Welcome to the Black Journey to Neuro keynote talk that will be given by Dr. Ishmail Abdus-Saboor. My name is Daria Lewis-Sanders, and I am an organizing member of BIN. First, to access the live captioning and the ASL, there's live captioning and American Sign Language interpreting available for this event. Captions, to turn your caption on or off, click the CC Live caption icon along the bottom of your window. And also to adjust the size of the font, click on the three dots at the bottom of your window and say more. And selecting subtitle settings is what'll get you there. Our sign language interpreters are Jamie and Catherine, and they should appear as panelists on your screen. If you experience any issues accessing the captions or interpreters, please message to all panelists in the chat. Before we begin, I want to acknowledge that I am joining from what we now know as Douglasville, Georgia, which are the lands of the Muskogee Nation. Since we are on this digital platform and joining from a variety of regions around the world, I really encourage you to reflect on the land that you are joining from. Please reflect on the relationship with that land, the indigenous peoples who have and continue to inhabit them. And please feel free to use the link that's in the chat at native-land.ca to find more information about the land where you settle and the people and the stories of that land. And as we roll the sponsorship, I want to thank our conference sponsors, including the Allen Institute, the Wu Tsai Neurosciences Institute at Stanford, and the University of Minnesota Department of Neuroscience, and Medical Discovery Team on Addiction, the Alzheimer's Association, Neurosity, and many more. Without their support, these events would not be possible. And I hope that you've been enjoying some of the programming over the past 3 days. And if you were unable to attend any of the sessions, please check out our YouTube page where all our events from today and the rest of Black and Neuro Week are either available now or will be posted shortly. And there's excellent resources there. So please check that out. Today's theme I'm very excited about is Black Journey to Neuro, and it's really all about embracing different trajectories and the many paths that we have taken within our BIN community. So in addition to this keynote, which you are about to hear, about an hour, well, really an hour at 2 p.m. Eastern time, we have an incredible virtual panel called Lessons Learned, Navigating Nonlinear Journeys in Neuro. And we have 3 incredible speakers representing different career paths and stages, and they will share their lived experiences and identify key lessons that they have learned during their journey. This panel will also provide an opportunity for open community dialogue to discuss tips for how to navigate different journeys in neuro, which we will end up turning this into a toolkit for you all. So we really hope to see you back for that. And throughout the day, we are also celebrating mentors who have played an important role in supporting us in our journey to neuro. So if you have a mentor that you want to shine a spotlight on, whether that be a teacher, or a supervisor, or a peer, please get on Twitter and Instagram and tell us who they are, why they are important, and we want you to use the hashtags BINW23, Black Journey to Neuro, and BINW23 Mentorship Spotlight that are here on the screen. All right, So I have the wonderful honor of introducing Dr. Ishmail Abdus-Saboor, who is an associate professor of biological sciences at the Zuckerman Mind Brain Behavior Institute. Dr. Abdus-Saboor attended an HBCU, North Carolina A&T, followed by a PhD in cell and molecular biology at UPenn and postdoc training at Cornell and UPenn. He has received many honors and awards, including an NIH K99 ROO Pathway to Independence Award and a BWF PDEP Fellowship as a postdoc. Since opening his lab, he has been named a Rita Allen Foundation Scholar, Sloan Research Fellow, Pew Biomedical Scholar, HHMI Freeman Hrabowski Scholar, McKnight Scholar, and a recipient of the Chan Zuckerberg Science Diversity Leadership Award and an NIH DPT New Innovator Award. Dr. Abdus-Saboor will tell us all about this journey in his talk entitled Fun Times in Neuroscience, which will be followed by an audience Q&A, and we invite you to write your questions in the Q&A box on your screen, and we'll try to get to as many as possible. Additionally, if you want to share any highlights from what you have learned today on social media, please do so using the hashtag BINW23 and Black Journey to Neuro. And without further ado, I am handing the mic to Dr. Abdus-Saboor.
Can you all see my screen? Yes?
Yes.
Great.
Yes.
Great. Yeah, so thanks, Dario, for that really kind introduction, and thanks to the organizers for having me here today. It's a real honor for me to be here with you all to address you. And I'm a really big fan of the community that you all built. I'm really proud that you all built this vital community for us. And I can't wait to see the future of Black and Neuro, and you all have my unwavering long-term support. So I was quite intentional in naming this address today, Fun Times in Neuroscience, because what I really want to convey to you all today is how much fun I've had and continue to have in neuroscience. And I'm really big on following your passion and doing the things that bring you joy in life. And certainly science and neuroscience is that for me. I feel immensely privileged to be able to, sit around and think about big problems and innovative ways to try to approach those problems and mentor and train the next generation of scientists along the way. So I hope I convey that to you, how much fun I'm having in this business. So I'll, over the next 35 to 40 minutes or so, I'll split the talk. The first half will kind of be all about my life and some key points that have gotten me to where I am today. And then in the second part, I'll tell you a little bit about some of the science that's ongoing in my lab. All right, so born and raised in Philadelphia. I guess my family has been in the Philadelphia section in the Northeast of USA for 3 or 4 generations. And before that, both sides of my family actually hail from South Carolina. So as a kid, I collected all kinds of pets growing up: turtles, snakes, lizards, dogs and cats, fish, etc. So I was really fascinated by biological systems and animals and thinking about, how things work. Science was certainly always my favorite class in school. I did well in all my studies, but certainly it was science class that got me really animated and overjoyed to be in school. And I remember a particular teacher, Mr. Moore, a 3rd grade teacher of mine at the John B. Kelly School in Philly. He's African-American male. And Mr. Moore took us on class trips in Philly and outside of Philly. We'd go on these long nature walks. He was overturning rocks and pointing out animals and plants. And I just derived so much joy from this. So definitely wanted to shout him out as being a vital part of my development and just, enthralling me with this love for science. So this was further solidified, I would say, in high school. So I attended Central High School in Philadelphia. It's actually the second oldest public high school in America, founded in the 1830s. It's one of these special admissions magnet high schools, and I was fortunate to be selected to attend here based off my track record at the time. And at the school, I was able to do a honors biology course my freshman year. So it was a year-long course, and it also entailed that we conduct a science fair project. So I was able to actually do this at home. So at age 14, as a 9th grader, I set up a lab at home. And, my parents allowed me to do this, very supportive of me. And my model organism at the time were these crayfish. And I was investigating regeneration rates in crayfish. And what the idea being is you sever one of their appendages, their legs can regrow. And I was testing a popular herbal supplement at the time, ginseng, and whether just spiking their drinking water with ginseng could speed up their rates of regeneration. I don't quite remember the results of that research, but I do remember it was the first time, I became a scientist. I had a lab notebook and I was recording my results and observations and presented a poster. I actually won a citywide science fair award for this research and was able to compete at the statewide level at Penn State University with kids across the state of Pennsylvania. So this early experience really solidified my desire and drive to be a scientist one day. I mean, it's the thing. Science has always captivated me and kept my attention. I've never wanted to do anything else in my life. I've never been as excited about anything else. And all right, so, after high school, where to go for college, I attended an HBCU, historically Black college or university, went to the South, attended North Carolina A&T State University because this school is known for having a strong background in science and engineering. And I attended an HBCU for a number of reasons, one of which, was just sort of following family tradition. So my parents attended HBCUs. They attended Howard University in Washington, D.C. I had a number of aunts also attend Howard. Had uncles attend Morgan State University and Virginia State University. My grandfather, the late Reverend John Parkinson, attended Lincoln University, which is the oldest HBCU in the nation. So I was surrounded in an environment with my parents and immediate relatives who, extolled the virtues of an HBCU education and convinced me that I should follow in this path as well and get a strong foundation here with people who love me and care about me and support me. And then I could, move forward in the world and go to, another institution for a graduate or medical school. And I'm glad I accepted their advice. It was the first time I got to meet other Black scientists. My parents have math backgrounds; they're not scientists, and it was important for me to be able to actualize myself and my future aspirations by seeing people who look like me and came from similar backgrounds, doing the sorts of things in life that I intended to do. So this was really important. And my peers and classmates, we were colleagues, not competitors. We were equally excited if a classmate of ours, aced an exam or landed an internship or got into a graduate program of their choice. It was like the same positivity had occurred to us. This was really important to have this environment where we really supported and nurtured each other and didn't see each other as competition. All right, so here I am. So the first lab I worked in was a C. elegans genetics lab, behavioral genetics with Dr. Werku. This is me, a recent homecoming, returning to the first lab I ever worked in. This was great for both me and Dr. Werku. All right, so leaving undergraduate, for graduate school, I went to UPenn and continued to work in C. elegans. I had an affinity towards this model system development in college. So I continued working with C. elegans, but here with a different set of problems. So my PhD is in cell and molecular biology. And here I studied the canonical RAS and Notch signal transduction pathways that are often oft used throughout development. And here I was studying how these commonly used signal transduction pathways control the cell fate and morphogenesis of these simple cell types in the worm equivalent of a kidney, the worm's primitive renal system. So just some things to note about my graduate career. So definitely there was a little bit of a culture shock in a number of ways. And as I've mentioned, in the HBCU environment, you're not a minority, you're a majority. And, we had this really supportive environment where, we all wanted to see each other succeed and win. It's a lot more competitive, at a place like UPenn. Definitely suffer bouts of imposter syndrome myself and some of the other Black and brown students. Graduate school is hard for everyone, but, when you're a minority in some, elite environments, if you struggle on an exam or you can't get certain experiments to work, especially those first few years, myself and others, we question ourselves and our abilities and, whether we were cut out to be at a place like this or whether they had lowered the standards to let us in, all things that are nonsense. But, definitely suffered these sorts of things early on, but having each other and supporting each other allowed us to persevere. And as I began to earn signs of success, this allowed me to increase my confidence. Papers being published, and I won a number of awards as a graduate student, and these things bolstered my confidence for sure. And by the time I ended graduate school, I definitely knew like academia is it for me. I want to run a lab one day. I was just so excited about thinking about experiments, designing experiments, going after big problems, and I knew it was time to do whatever it took to try to land a faculty job one day. So I just want to acknowledge these folks here. So I mentioned that the peer group I had in graduate school was vital. So these are, was no Black in Neuro back then, but maybe this was our local equivalent, the E-Just Biomedical Society. And all these people here were PhD students with me together at UPenn. They're all doing wonderful things now in academia and industry and government. I'll just give a shout out Dr. Latasha Crawford, who's here at the bottom right of the screen. We were just in a study section together last week. She also is a pain neurobiologist, a junior PI, done really well at Wisconsin-Madison. And so we supported each other socially, intellectually, academically. We did a lot of outreach activities together. I was actually the outreach chair for this society in grad school. One of the things we did, myself and some others, we started the Science Education Academy in West Philadelphia, where we would go on Saturdays to provide hands-on science demonstration to inner city kids in West Philly from grades 2 through 6 to kind of supplement what they were learning in their science curriculum. And we trained many students over the years, and proud to say that this organization is still running strong, 15 plus years later. Dr. Danielle Haney, who's here, is still running it now. All right. So as a postdoc, there were a number of big leaps for me. So I moved from genetics and molecular biology to neuroscience, and I moved into sensory neuroscience. I went from working with worms to work with mice, and again, this leap from molecular biology to sensory neuroscience. I did 2 postdoctoral fellowships. So the first, I worked with Ben Scheiken, and I was studying monoallelic expression of olfactory receptors and olfactory sensory neurons in the mouse nose. And the second postdoc, which is more closely related to what I do in my lab now, studying touch and pain, somatosensation also in the mouse. And, so why did I make this leap to neuroscience, having never, taken a neuroscience course in my life? It was because, if I was honest with myself, as I was concluding graduate school, the papers that I would read for fun, the talks that I would go to where my mind was just blown and amazement of what people were doing and some of the outstanding questions was all pointing me towards neuroscience. So I knew I had to make the leap, and I figured I could learn or teach myself the discipline as I go. And, definitely in the second postdoc lab, I was an experimental and, intellectual black sheep. And some of my mentors, I remember at the time, didn't think it was the wisest idea because my second postdoc lab was more of a developmental neuroscience lab. And I had all these ideas to do animal behavior and optogenetics and the peripheral nervous system and some other ideas and approaches that I wanted to get off the ground, but no one in that lab was working on it. So here I was, new animal system, new discipline, and I'm going to a lab and kind of working alone, essentially, on things that no one else in the lab is working on. But I'll say, things eventually worked, and it definitely, I wouldn't change it because it gave me confidence, especially opening a new lab. When you become a, excuse me, when you become a junior PI and you get a new lab, it's empty lab, and a lot of money, and you're told to, go do something important, and we'll check back in 5 to 7 years and see if you get to keep your job forever. So it's a daunting time for any new faculty member. But because I had this experience as a postdoc where I had to develop and set up something from scratch, it gave me the confidence that I can do this all over again in my own lab. So I think it paid off. and, published a number of papers as shown here, a number of grants like the Burroughs Wellcome Fund, PDEP, and NIH K99 R00. Those grants were important and set me up and did well on the academic job market and opened my lab about 5 years ago now, summer of 2018. So I don't want to present the scenario as if this were like a straightforward linear path and that I didn't have hurdles or challenges to overcome, especially as it relates to my Blackness. So I thought I'd just mention this 3 quick illustrative stories, which could be easier than speaking in the abstract. So I remember there was a time in graduate school where I was in the middle of campus and waiting for the campus shuttle to take me from campus to my apartment. And, the university police descended upon me, like 5 or 6 cop cars, and the officers jumped out and they're barking, yelling orders at me and asked me to produce ID. And they told me I fit the description of a criminal or suspect they were looking for. And I remember, my hands are shaking, and I took out my wallet and showed them my PIN ID. And, they kind of looked in astonishment that I was a student, and they just handed my ID back to me and wrote off, no apology or no nothing, and I just, had to go get on with life. This was maybe as a 3rd or 4th year graduate student. I remember another incident as a postdoc. I had won a fellowship, and I guess the email just came in, and I was like super excited and talking about it in the lab, and people were kind of coming over to see what the commotion was, and people were congratulating me and things of that nature. And then there was another student who came by and said, "Oh, what's that? You won one of those minority things?" And I still remember that to this day, right? Because the attempt was like to, stick deflate my balloon, right? To an attempt to steal my joy, right? And it stung, right? These things hurt because we know that, oftentimes we have to work 2 or 3 times as hard as others, and no one's given us anything. Right. Even if something may be earmarked to support minority students, they're not giving them away. They're very hard to win. I've actually done better with awards that are for everyone than ones that are for minorities, because they might not even be as much funding. So no one's handing things to us. So but these things hurt when people do this, and it's an attempt to steal our joy. And, faculty, I remember, maybe foolhardily thinking that, you rise as you're rising the totem pole from graduate school to postdoc, and now you're a faculty member, you're at the top of the food chain, and certain things will stop happening to you, but that's not necessarily the case. I remember, times going into my own building where I worked, and people stopping me, asking me to show ID, and what am I doing here? And you don't belong, people who are not security, right? And these things hurt, and they're challenging for sure. Now, as I present these things, I don't want to be disingenuous and present a picture as if this is my experience in academia, because it's not. As I mentioned, I'm having a lot of fun. These are relatively isolated events, and for the most part, my days are filled with fun and joy in science. But these things also happen too, and they're real, and they hurt, and they're challenging. But how do I overcome these things? The session yesterday on neuro-racism and mental health was really good, and they talked about some of these sorts of discrimination and racist events and coping strategies, and it really mirrors some of the things that helped me get through. Definitely having like wonderful mentors and a community, people who I could turn to when these isolated events happen, to kind of support me and encourage me, and just a shoulder to lean on and cry on. Family is very important for me. Confidence, once I'm at a point where my confidence is really high and belief in myself, that when these sorts of things happen, they don't derail you as much. I'm a person of faith and definitely rely upon that. And that's been really important for me. And purpose, I feel like a strong sense of, this is what I was born to do. I'm probably not too good at anything else, but I definitely feel like committed to this lifestyle and this career, and doing important science, making important discoveries, and training people. And I won't let anyone derail me from that from my purpose. So I talked about mentors. Let me just highlight some of them now as I wrap up and get ready to move over to science. So here are my three primary mentors, my PhD advisor and two postdoc advisors. And they were all wonderful, but, ran distinct labs. But I think there are certainly some commonalities or shared features that allow me to be successful, especially as a Black neuroscientist in these labs. So all of my mentors had an understanding of what it's like to be Black in America, cultural competency, I would say. And really importantly, they treated me like everybody else, meaning they pushed me, they challenged me, they gave me the toughest projects that they would give to anyone else. They had high expectations of me. Sometimes I've heard some mentors say, they've never mentored a Black scientist, so they don't know how to treat us. They don't want to be too hard because they don't want to be seen as racist or too soft, or they're very confused. But what we want is to be treated like everyone else, the same resources you give to other people, the same intentionality and input. Give it to us just like that, while also having some cultural competency about who we are and things we may be experiencing. And I definitely got this out of my mentors. A champion for my career goals for sure, and long-term support. I still engage with them. They're still writing rec letters for me, and I hope that these are lifelong friendships at this point. I have a whole host of unofficial mentors too, and I think that's really important. I have a mentoring village. I have the same shirt on now. Here's another picture with this shirt on. I like this shirt. Here's Mikey Nussbaum, who is at UPenn, and he, as my 4 years as a postdoc at UPenn, I probably met with him every other week or so to talk about science, career, life things. He actually did a year-long one-on-one tutorial with me, just me and him in his office teaching me neuroscience. I mean, Mikey has literally taken the tie-dye shirt off his back for me. So much I could say about Mikey. More recently, some mentors here at Columbia, Nobel laureate Richard Axel and illustrious Charles Zucker. I have regular science meetings with them. We talk about ideas and projects going on in the lab, and that's a lot of fun. Other mentors here at Columbia, and all these people I meet with on a regular basis, lunch, dinner. It was mentioned that I've been successful with a lot of awards and things and papers, and these folks are reading everything that I put out there and editing and commenting and pushing me. So it's really great to have a team of really supportive people. And one thing that I'll mention, which you can clearly see phenotypically, is that none of these people look like me or come from a similar background. And I think that's okay. I do have lots of mentors that do look like me, and we can connect on some other levels, but I found throughout my career, there are lots of wonderful people who want to support us and mentor us and serve as allies and supporters. You just have to find these people and don't necessarily judge a book by its cover or write people off because they may not share your background. So these are mentors that, I'm kind of looking up towards. These people have run labs for 30 to 40, 50 years. But I also think it's important to have people who are, at your similar level. So I'll just shout out Dr. Kav Zarasa from Duke University. Kav is a few years ahead of me. I could spend the next 30 minutes talking about Kav's impact on me and our entire community. Just him being, his presence, for me has been so important and critical, just to see another Black man in neuroscience who has achieved so much, because it gave me someone to emulate. It gave me, I could say, okay, here's where I could be in a few years if I do some of the things that Kaf has done. And alongside that, again, he's been here to read my papers and grants and to strategize, and a shoulder to cry on, and I can go on. And he doesn't just do this for me, he does this for lots of us in the community. So his impact is just exponential. Speaking of peer mentors, I like to highlight the brilliant Dr. Bianca Jones-Marlin, who's my neighbor here at Columbia. It's so great being in her company. This is us recently at HHMI headquarters, where we both were awarded a really prestigious grant and funding support. And we worked together on this for months together. knowing that we were both, competing with one another, but we didn't look at it that way. We were like, let's both get it and let's support each other together. We worked together for months, and glad that we both got it. And I'll just highlight one of my best friends, Dr. Ishmail Ahmed, who is a senior postdoc. So I think it's important too to have mentors who may be even a level below you, right? We get together, he and I, and we talk science for hours. We get lost in ideas and experiments. It's so fun. And he advises me. I ask him so many questions, and I learn a lot from him, and just love his companionship. And just lastly, I'll just mention my family. Family is very important. So here's my lovely wife of 14 years now. She's my confidant, best friend, supporter, been with me every step of the way, and I couldn't have done it without her. Here is our lovely family, our two kids. We were on vacation last week. Here's that darn shirt again. My goodness. I will say this is my wife's favorite shirt, so I'm forced to wear it weekly. And here are my lovely parents, my mom and dad. I mentioned they attended Howard University. My dad was a math major and has worked as an actuary, and my mother was an accounting major, has worked as a CPA, and actually she's now chief financial officer of a major company. So definitely have highly successful parents, and professionals who definitely extol the virtues of education and hard work. And my parents are just the most wonderful people. They're sweet, they're kind, caring. They're just like me. They're very soft and humble and soft-spoken but brilliant. And I am the living embodiment of my parents, both nature and nurture wise, and everything that I am today is because of them. All right, so now let me switch over to the science that we work on. So my lab is very interested in how our brain perceives, generates the perception of pain and pleasure based off sensory stimuli applied to our skin. And we really like to integrate the peripheral and central nervous systems as we think about these problems. So over the past 5 years, we've been kind of delineated into thinking about both touch and pain. So on the touch side of the lab, we've been identifying these pathways from the skin to the brain that are really important for rewarding touch. And this is some of the work that I'll briefly highlight in a minute. And we've also been studying pain. And our work on pain, we've gone further upstream and just asked the question, if you're going to use rodents to study pain, you need to be able to accurately approximate their pain states. And we've developed systems to go after this using new forms of videography, machine learning, and we've developed our own in-house software packages to allow us to more accurately measure this hard to articulate sensation, especially in a nonverbal animal. But just for the sake of brevity, and I'll focus on our touch work. So to give credit where credit is due, Team Pleasurable Touch, these were some brave folks. They showed up right after the lab opened and decided to work on something that was new for all of us, and just kind of see where it took us. So the work was led by Leah, who's now a Jane Coffin Child postdoc at Hopkins, and Melanie's now off to industry. Two amazing technicians worked on this project, Bella and Will. I didn't do a good job of scaring them away enough. So they came here to Columbia for graduate school. Bella just joined the lab and is already off to some really cool stuff, following up on what I'll show you now. And Will's doing a third rotation here at Columbia, and we'll see if he decides to come back as well. All right, so gentle stroking of the skin is rewarding to mammals. I think we all would appreciate that. It's like we're hardwired for this sensation. It feels good and activates the activation of these neurons in the periphery, triggers circuits in the brain to generate reward and learning, so that we desire and crave this sensation. So it turns out many species, have these mammals have these neurons in the skin called CLTMRs, let's say, that are tuned towards gentle stroking detection. They were first identified in cats in the late 1930s, where investigators could do experiments like this, where you can stick an electrode in a forearm, and they could record units that they term, tickling. They were identified in humans in 1990, where you could do psychophysics experiments combined with electrophysiology. So you could stick a forearm in a person's, an electrode in a person's forearm, and that simultaneously stroke with cotton wool at a slow stroking velocity. Ask the person, how does this feel? They say, oh, this feels wonderful. And they found neurons that fired towards that stroking touch. And importantly, those same neurons did not fire to pain or temperature or other sensory stimuli. So neurons tuned towards like detecting socially relevant cues. So in the skin. So these two papers from David Anderson's lab came out I read them when I was a postdoc, and my mind was just blown. It was just, fascinated. I remember at the, Ishmail Ahmed, who I talked to, we would just spend hours just talking about these papers and dreaming up experiments and ideas. And, these papers were important because the Anderson lab at Caltech had identified a putative population of sensory neurons in the mouse that shared similarities to these CLTMR neurons. They were labeled by a single gene called MRGPRB4, a G protein coupled receptor. And they were able to show, just like I showed you in the human data, that they put the recorded calcium neuronal activity via these calcium transients with the genetically encoded calcium indicator GCAM, and to stroke the flank of the animal that these neurons fire. So very similar to what's been shown in other species. So, again, these experiments, I thought were just so striking. And I thought there were, lots of exciting experiments and ideas to move forward with this, two of which we had were this. One, we wanted to ask the question, could these neurons be involved in any natural, ethologically relevant social touch behaviors? And then two, if this is rewarding touch, how does this engage with reward circuits in the brain? So this was sort of our point of departure in the project. So we reasoned that, if we wanted to be able to implicate these neurons in any natural behaviors, then we decided to take a different approach, an optogenetic approach, but maybe a bit of a play on optogenetics. So as this community, I'm certain, is aware of, optogenetics makes use of the blue light sensitive ion channel, channelrhodopsin, but most neuroscientists activate neurons in the brain. They'll cannulate an animal to turn on or off neural circuits as animals behave. We reasoned that, what if we put channelrhodopsin not in the brain, but in the skin into these neurons using mouse genetics? And if we were able to do this and focally shine blue lights, spots of blue light to the skin of a behaving animal, might this mimic touch amongst animals? So this was our strategy that we decided to take. So here's just our Crelox genetics showing that we put channelrhodopsin in these B4 lineage neurons. And here's these beautiful sensory neurons in the skin of these transgenic mice. You can see they're associated with the hair follicles. And the reason why the experiment works that I'll show you in the next slide is here. Hopefully you all can see my cursor. The blue light can penetrate through the skin and directly hit these sensory terminals to activate these neurons in vivo. We can do experiments like this, which is just sort of a sanity check, where we can just acutely dissociate these B4 lineage neurons, and we applied mechanosensory poke to these neurons, and we could show that indeed we get these receptor potentials, so they are indeed mechanosensitive, but not in B4 negative cells. And then in collaborative experiments with a great colleague and friend, Vicky Abrera's lab at Rutgers, we could do spinal cord slice physiology, where we optically activate these B4 terminals and then record light-induced currents downstream in the spinal cord. And you can see we get these light-induced currents. So this entire slide is a sort of a proof of principle to show that we do have a system whereby we can activate these touch neurons in vivo. And here's what we saw that really gave this project wings and really excited us. So here on the left is a control animal, and you can see we're pulsing blue light to its shaven back skin. Animal realizes that the experimenter is in the room, but doesn't really alter its behavior. But look at the experimental animal on the right. You can see this animal is behaving as if it's been touched or stroked on its back. Some of you have cats, maybe you touch your cat on the back, it kind of adopts this posture. Animals not running away from the light. We don't think this is a pain or aversion response. I won't show you today, but we've done the same thing with other pain or itch optogenetic lines. And in those contexts, the animals run away from the light. They're not running away, but this, again, as you can see, this doing that lowering posture. We can quantify this behavior just by how low the animals do this back dip, and we see a nice statistical separation between our experimental versus control mice. We don't see this in a very related population of neurons, the MRGPRD neurons. So something specific about this B4 lineage. So next, to get at valence of how the animals may be perceiving this, we developed this this peripheral optogenetic condition place preference, where we have a number of conditioning stimuli on each side of the chambers. But importantly, whenever the animals are on one side, they get this blue laser light. And whenever they're on the other side, they get control green laser light, which the animals can see just as well as blue light, but it won't activate the channel diopsin. So it serves as a nice control for us. So we do this for 5 days. Then on the 6th day, we just remove the lasers and ask, where's the animals prefer to spend time? And indeed, they prefer to return to the environments where they were previously optogenetically these B4 neurons were optogenetically activated. And we do not see this with the related MRGD population. Okay. So it appears that the animals find this activation rewarding. So at this point, we had this posture, and it seems like the animals may like it, but it didn't get back to our original question of whether this could be a natural behavior. So we thought a lot about what this posture looks like, and we settled on lordosis, which is the female sexual receptivity posture. And we actually began all these experiments in female mice. So this is also why we thought this could be lordosis, where the animals adopt this back-lowering posture. Lordosis had been studied for a number of years, since the 70s, by many groups, but mainly from the context of hormonal and olfactory cues. Certainly, there's a lot of touch that occurs during mouse mating, but no one had identified the mechanosensory touch neurons that may be involved. And we thought, maybe our neurons are involved in this posture, this behavior somehow. Maybe with our optogenetic stimulation, we're bypassing some of the needs of the mouse and kind of mimicking the male touch. So if some of those ideas are true, how can we prove that? So simply, we just decided to genetically ablate these neurons, okay, using, again, Krelok genetics. And you can see here, we ablated these neurons, but kept the other population, these V1 neurons are intact. And to look at mating, we adopted these lordosis quotient assays, which are well performed in this mating literature. So we needed to overreactimize the females to remove their endogenous source of estrogen, and then we control when the females are in behavioral estrus. And then we did all these experiments at night when male mice mount. And here was our regimen for these experiments. And importantly, we did this over the course of multiple weeks, because there's literature showing that female rodents increase their sexual receptivity over time. Presumably the female is learning how to perform this behavior, and there's a reward learning component, so it goes up over time. And so this is why we adopted this behavioral protocol. So in wild type females, we essentially recapitulate published data where the female sexual receptivity is here and it goes up over time. We see the exact opposite when we ablated those touch neurons. They actually start at a similar level of sexual receptivity as wild type females, but by the 2nd and 3rd week, it's dramatically reduced to the point many are no longer sexually receptive. So almost all wild type females are going up in sexual receptivity, and almost all these touch neuron ablated females are going down in sexual receptivity, shown this way as well. What are these females doing when they're no longer sexually receptive? They actually become quite aggressive to the male attempts to mount, as shown here. And this is something that you almost never see in a female that is sexually primed. So what these data show us are that these B4 lineage neurons are critical for female sexual receptivity. But we think it's not just for the sense of touch, because this is a very small population, 5 to 10% of all DRG neurons. There are lots of other touch neurons intact. There are piezo2 neurons, TrkB, RET positive, et cetera. So these females can sense touch, but what we think is that these neurons in the skin are activating or turning on critical reward circuits in the brain, such that if you don't have that signal in the skin to the brain, then what is this experience like to the female? Maybe this feels like a male touching them, but there's no rewarding component. So if that idea is true, how can we prove it? So as I wind up, I'll just show you some of the experiments we did to try to get at that question. So first, we just asked a sufficiency question. Does turning on these neurons in the skin engage reward circuitry? So we generated another mouse line that would allow us to combine the skin optogenetics with brain recordings in the nucleus accumbens, a well-known reward center. And here we would measure the neuromodulator dopamine with the high affinity dopamine sensor. And when dopamine binds this GRAB, sensor, it emits photons of light that we can detect with a photodetector and read in a computer. And I'll just say at this point, with this project, again, this started as a side project, and it took us in a number of interesting directions, which also means that the grad students and technicians who worked on this, we had to kind of develop and invent these strategies from scratch, because no one had done a number of these experiments, including us. So very, very heroic team of people worked on this. Just want to give them their credit and how hard they worked to design some of these things. All right, so you can see that stimulation of these neurons in the back skin led to this dopamine release. We also saw dopamine release when we shine the light to the anogenital skin, where these neurons reside and where there's a lot of touch during mating. This also gives us this dopamine release. So turning on these neurons in the skin is sufficient to engage this canonical reward signature. How might, I'm actually going to skip over this part for the sake of time and just go to the schematic. So how might you get from the neurons in the skin to the brain? So what I just skipped over for the sake of time was a number of interrograde and retrograde tracing experiments and some electrophysiology for us to kind of come up with a putative circuit for how you can get from the skin to the reward center. It looks like you can do this in about 3 to 4 synapses. Here are B4 neurons, and downstream are these GPR83 positive neurons that go from the spinal cord to the parabrachial nucleus, and then other neurons go from the PBN to the VTA to NAP. Okay, this was from a dispatch article from another group who independently wrote about our study, and I thought this was a really nice overview and schematic of what we discovered. So and finally, we showed that loss of dopamine release is lost during mating in the absence of those touch neurons. So we went back to our photometry recordings, and you can see in a wild-type female, at the moment of male mount onset, we see this nice dopamine release, but this is lost when these touch neurons are ablated and quantified there. So this does provide an explanation to the behavioral lack of receptivity. The animals don't engage anymore, likely because it's not rewarding. And this finally, as part of some ongoing experiments, we asked the question, what about individual VTA dopamine neurons? So these experiments, we were looking at bulk dopamine release, but we went back to the VTA to ask the question, which is still an ongoing question for us, could any of these VTA dopamine neurons be tuned towards rewarding touch? Maybe not other stimuli. So to do this, we use miniature microscopes and endoscopics, and we put the Genelia GCamp virus into dopamine neurons and do these recordings during the context of mating assays. And we aligned moments of anogenital sniffing or mounting, the touch that occurs during these behaviors, with calcium activity. And indeed, we could find some cells that seem to be active during the touch during these behaviors and not during other moments. So consistent with our idea that maybe some VTA dopamine neurons are tuned towards rewarding touch. And this is an idea we are continuing to explore. All right, so we published this paper earlier in the year, and just some highlights again. So this focal activation of these neurons leads to that social posture. And I think the exciting thing too is that this can occur even in a non-social setting, even with no other animal there or no other sensory cues. So it shows the importance of these neurons for engaging or turning on social behaviors. And also, I think it points towards the potential of even targeting the peripheral nervous system to enhance social reward. genetic ablation of those neurons, we saw a reduction of touch-dependent social behaviors. Here we focus on mating. However, these neurons tile everywhere in the mouse where there's hairy skin, leading us to believe that there are many other behaviors that may rely upon these neurons. And we have some exciting preliminary data that looks like we found multiple social touch behaviors that do require these neurons. And some ongoing work our lab has in collaboration with Rainbow Holtman's lab at University of Iowa. We show, and this work was led by a former graduate student, Melanie Schaffler, in the lab, that ablating these neurons in early life seems to be like an early life stress model, because the animals grew up to have problems with stress resilience. Sasha Fulton, who's a new postdoc in the lab, is following up in this project and adding a lot more data. And we're still wrapping up this paper and adding a lot more. So look forward to this next year. I think this is another cool direction. Recently, we've collaborated with Catherine Dulock's lab at Harvard, and they have a really nice social homeostasis paradigm where if you isolate animals for a number of days and then there's a reunion, the animals have a desire to spend more time with the animals that they were previously with, to get back to a sort of social homeostasis set point. And we were able to show together that without these B4 neurons, that social homeostasis behavior is lost, that rebound is lost. So yet another social context that requires these neurons. And I'll just say, as I mentioned, it started as sort of a side project, and it led us in these unexpected directions, and the lab is growing, and amazing people, and there are 5 new projects that have spurred that are ongoing from this one paper. So if you all invite me back one day, I can talk maybe about some of these other projects. So with that, I'll conclude. This is a relatively recent photo of the folks in the lab. Really fortunate to have a number of organizations invest in our work over these 5 years, and happy to take some questions if you have them. Thank you.
Thank you so much, Dr. Abdus-Saboor. And we do have questions. And as a reminder, you can put your questions in the Q&A. The first question, starting with the statement that it was truly amazing and your work is so fascinating, and thank you for sharing your journey, followed by a question, what about disorders where touch, specifically soft touch, is no longer rewarding but noxious? Do you think that there is rewiring or impairment of which level in the circuit? I actually had that question too.
Yeah, it's a great question. There have been, a handful of pioneering studies from David Gintes and Lauren Orefici at Harvard, where they've showed, quite interestingly, looking at in autism models and looking at high confidence autism risk genes, that even ablating or manipulating these genes in the periphery is sufficient to engage autistic-like behaviors, where you have these tactile hypersensitivity, and in some of their work they've shown that this is local in the DRG and spinal cord. So I think for sure, but looking at the peripheral nervous system for some of these disorders is in its infancy. People have mainly looked in the brain. Again, I keep saying, the untapped potential of the peripheral nervous system. I think as we begin, our lab and others begin to look in the periphery and spinal cord, we will see instances of rewiring or plasticity that may underlie some tactile hypersensitivity. Just before this Zoom call, was meeting with one of the students in the lab, showing me some data where we think we found a tactile hypersensitivity phenotype and some alteration, plasticity in the DRG. So I think it's quite exciting times to put the microscope under the periphery and not always under the brain, or only under the brain, to look at disorders where touch may be altered.
Thank you. Another question, do you see the B4 lineage rewarding touch in males as well as females? And does it also affect mating in males?
Yeah, that's a great question. That was the one common question that all 3 reviewers asked us. How does this look in males? When people start experiments in males, no one ever asks, how's this look in females? But in males, It's unclear, I would say. So for one, the gene expression of the B4 gene is shared between males and females. We don't see any sort of upregulation in females. When we did the blue light back stimulation, quite interestingly, we do see that back lowering posture in males. We don't quite know what that means. Males have a whole constellation of behaviors. Sometimes male mice mount other males, and as a sign of like submissiveness, the other male kind of lowers. So could it be a behavior like that? We don't know. I also say we don't know because when we did the condition place preference in males, there was no preference or aversion, whereas we did see the preference in females. We didn't do the brain recordings in males, so it's unclear. But what I might speculate is that the circuits, the local reflex circuits are shared in the DRG, in the peripheral nervous system in the spinal cord, but perhaps there is sexual dimorphism with this ascending circuit as you get to the brain. And where maybe there are more potent inputs to reward centers in females than males, but that's speculative at this point. That's a great question.
And then relating this mouse work back to humans, is this a general rewarding touch on skin or erogenous zones?
Yeah, that's a wonderful question that we are following up with now. As I mentioned, these neurons tile everywhere where there is hairy skin in the mouse, but it does seem like there may be hot spots in the skin, like the back or genitalia. That dopamine response I showed you, we got more dopamine release when we turn on the neurons in the genitalia versus in the back. So we think there are hot spots, and how do you even wire a system that way? It doesn't look like we see stronger innervation of the sensory neurons in certain areas in the body, but probably the ascending circuits to the brain may be altered. So Isabella Sushi, who's a grad student in the lab, I think that's going to be her thesis project. She was the second author on the recent paper, and she's designing experiments now where we can, in collaboration with Itamar Khan's lab here at Zuckerman, where we can do our peripheral optostimulation in a hit fix preparation. So we have really nice temporal spatial control over the animal, and we'll literally shine the spots of blue light everywhere in the body and record dopamine release, and while simultaneously doing a wake fMRI to get brain-wide connectivity. It's a crazy experiment that's never been done, but if it works, I think it can address that question that was raised.
That's incredible. And then the last question that we have is, I lost one. Is, do you think activating B4 can reverse the negative effect related to noxious touch, like as an analgesic therapy?
Yeah, check back with Dr. Sasha Fulton in another year. She's designing experiments to address that question right now.
Yeah, and then one final comment. People are asking if there's any way to contribute to your lab. Are you looking for anyone to contribute to your lab?
We're always looking for bright and inquisitive and fun people. We like smart people and we like good people. People who get along well with others and collaborative and like to have fun. And lab is kind of growing, and but reach out for sure.
Well, thank you so much for your time, and thank you everyone for coming. And again, I want to thank our conference sponsors. Also, a big thank you to our live captioners. And as a reminder, all of our events are available on our YouTube page. If you haven't yet, join the conversation on social media, hashtag Black Journey to Neuro. We have BINW23 mentorship spotlight, so please spotlight the mentors that allowed your journey to be possible. And we're at Black in Neuro across all platforms. And also, for the most up-to-date information on the conference, check out the conference booklet on our website. And we hope to see you this afternoon for the rest of our events for today and for the rest of the week. Thank you so much for coming.